This video is sponsored by the LMU Biology Department. You all know about these different animals such as the sea star and fish, but have you heard of this one before? Well, it's the Trichoplax adherens, the only formally described species in the taxa Placozoa. In the 19th century, a zoologist by the name of Franz Schulze found an amoeba-like animal crawling on the side of an aquarium at Graz University. But now, they are known to be found in tropical areas with temperatures of around 32 degrees Celsius and in highly diverse ecological systems like rocky pools. Despite knowing where they can be found, most of what's been learned about their biology has come from studying them in laboratories around the world. Now, the Placozoa are described to be very simple animals, and they even have the smallest amount of DNA ever measured for any animal, with around 98 million base pairs, which is minuscule, especially when comparing it to humans who have around 3 billion base pairs. With this, their body has been described to be transparent, flat, and small, with no presence of organs, specialized nerves, or even muscular cells. Because of this, their body has no symmetry, is irregular, and changes constantly. Their body, however, is made up of a few thousand cells of just 4 types, which is a small amount when comparing it to sponges who have around 10 to 20 different types. They have 2 distinct tissue layers called the ectoderm and endoderm, where its ventral side is made up of column-shaped cylinder cells and gland cells, while its dorsal side is composed of cover cells that are ciliated and flattened towards the outside of the animal. Lastly, there are also fiber cells found in the space between these 2 tissue layers. So, how do these Placozoa move around? Well, they move in 3 different ways. First, they move with a flagella, which is a membrane-bound whip-like structure that helps it produce a force on the water around it. The second is that they use their cilia, which is a bunch of finger-like projections that displaces the water around it. And lastly, they use amoeboid movement. This is where they change the shape of their body in order to move. And these motions help them feed with their ventral side, by forming a digestive cavity and releasing enzymes around their food. But since they are kept in labs, scientists aren't sure what they actually eat, but they are kept alive by feeding them chromis and chlorophytes, which is also known as green algae. And aside from feeding, they have been found to reproduce both asexually and sexually, based on environmental factors. So they reproduce asexually when population density is low, via binary fission and sometimes budding, and they reproduce sexually when population density is high. But their bodies have to degenerate, so something like an egg or a sperm can develop in the interspace of the placozoan. Despite observing their reproduction, knowing their developmental patterns has been so difficult to find. The scientists only know their development until the 64th cell stage, and since they live in laboratories, they have been hypothesized to live infinitely. With our information on the placozoans, 2 phylogenetic trees were produced in relation to the 7 phyla that were discussed in lab, where one tree was made from 16 sRNA data, and the second tree was based on the data matrices morphological characteristics of these organisms, with 2 added characteristics of eye-type and sexual dimorphism. So in this tree, the placozoans' closest relatives were the Echinodermata and the Chordata phyla, while its closest relatives on this tree are the Arthropoda, Chordata, Echinodermata, Annelida, Cnidaria, and Mollusca phyla. It's good to note, however, that these trees would change by being more general or specific about the data that was used to make the trees. And comparing our trees to the tree in the concatenated analysis sheds light on early metazoan evolution and fuels a modern Metazoan hypothesis paper. They were considerably different. This may be because the 3 trees were constructed with different data, where in the paper they used 17 characters, mitochondrial ribosomal sequences, and nuclear coding genes of 24 taxa, while our tree only used 16S data and morphological data of only 8 taxa, which may contribute to our inaccurate phylogeny. For the positions of the Placozoa, researchers weren't able to find an exact origin, but it states the Placozoa is a very difficult taxa to place on a tree, and that has even been seen as a sister taxa to the groups of Cnidaria, Cnidifera, and Bilateria. But recent studies show low support for any of these hypotheses, and states Most of these are due to the problems of molecular systematic analyses of these large data sets, because it makes it so hard to place the Placozoa on a phylogenetic tree. So since scientists don't know much about the Placozoa, more research needs to be done in laboratories around the world, so we can really understand this unique taxa, even more than we already do.